Stomata are microscopically small but extremely important structures on leaf surfaces that consist of a pore surrounded by a pair of specialized cells called guard cells. Their number, size, and location vary across plant species and can be influenced by a variety of environmental factors. To understand this variability, you need to be able to find and measure stomata, but even under the microscope they are not easy to see on a leaf directly. So you need to take a more indirect approach. This is a fairly straightforward procedure. You'll need clear nail polish, clear packing tape, scissors, microscope slides, a pencil or pen to write on the slides, plus of course a microscope to view the finished product. Later, you'll also make brief use of a stage micrometer. Paint a swath of the nail polish on either the upper or underside of your leaf, depending on your project. Try to avoid the major veins if possible, as they can make the polish harder to deal with later. Let the nail polish patch dry for a couple of hours. Putting the leaf under a hood can shorten the drying process down to 15 minutes or so. Once the nail patch is dried, cover it completely with a piece of tape. Press down gently but firmly. Carefully peel the tape off the leaf. If the nail polish patch is dry, the tape should lift the patch off with it. You will be able to see the patch as a cloudy impression on the tape. Stick the tape onto a microscope slide. Use a pen or pencil to write some sort of ID code signifying the leaf's species, the treatment group if any, and a unique specimen number directly on the slide. Using a microscope, look at the nail polish under 40x magnification. What will you see? Well, hopefully your nail polish has formed a perfect and transparent impression of the surface of the leaf, in which you can now see stomata. Move the slide around to find an area that looks like it has a high density of stomata. Stay away from the edge of the impression and avoid major leaf veins, dirt, and damaged areas. Count all the stomata that you see. Be sure to follow a consistent rule about counting stomata that are only partially visible along the edge of the field of view. For example, you might only count stomata that are at least halfway visible. Move the slide to two other high-density areas and count their visible stomata. The highest of the three stomatal counts will be your one data point from this impression. Repeat this process with as many leaves as necessary to build up a sample size appropriate for your particular project. This procedure usually works well, but some leaves have extra surface coverings that can obstruct your view of the stomata. These can be hairs, a waxy coating, or glands. In addition, the chemical solvents in nail polish can damage the leaves of sensitive plant species, preventing the formation of a usable impression. One interesting possibility is to use non-toxic, water-based varnishes, which could also allow you to get stomatal peels from intact leaves on plants in the field. Microscopes can vary in the precise size of the field of view, so you'll need to convert number of stomata at 40x to number of stomata per millimeter squared. This is where the stage micrometer comes in handy. Center the scale on the stage micrometer slide under the microscope using the same magnification you used to measure the stomata. Each of the smallest lines are 1/100th of a millimeter, or only 10 microns apart. Measure the diameter of your field of view by carefully counting the number of lines across its maximum width. This is not how one usually finds the scale for an objective lens, but our microscopes do not have the corresponding eyepiece scales called ocular micrometers, so we'll have to use this less precise approach. Now you can calculate the area of the field of view using the familiar formula: the area of a circle equals pi times half the diameter squared. Finally, to calculate the number of stomata per millimeter squared in our leaf, we simply divide the number of stomata we saw by the area of the field of view. This simple technique can provide powerful insights into how plants interact with a changeable and often hostile environment to access and protect the resources they need.